All right, so welcome to the lectures on meiosis and sexual reproduction. So when we talk about meiosis, it is the process of making an egg or sperm cell. So this happens in lots of animals and plants, sometimes fungi, some of the protists. So not all organisms use this method of reproduction. What it takes is specialized germ cells, not cells that make you sick. Germ cells is the term used for the group of cells that will go through the process of meiosis and then enable the organism to reproduce sexually. So you look at the dragonflies or the lions and the tortoises, and then you have the bee, but you also have the flower. All of those organisms are producing eggs and sperm. In the plant world, we call it pollen. Same stuff. In the animal world, we call it eggs and sperm. But they're making those specialized cells that enable reproduction. And it takes one egg and one sperm to combine to then produce the new offspring. Okay, so that's what meiosis is all about. It happens all the time. We see animals and plants and organisms doing this all the time. This was something I discovered years ago when I came home one evening, 2 slugs reproducing on my front porch. Now, the interesting thing here with the slugs, they're hermaphroditic. It means they have both male and female parts. So one slug is going to pass sperm to the other, and then it's also going to receive sperm. So being a hermaphrodite, you actually double your reproductive opportunities, double your reproductive potential. Okay, but when we look at meiosis, check out these 2 dogs. These are Rhodesian Ridgebacks. They are both going through meiosis. The male, as he goes through meiosis, the end result is the production of sperm in males and eggs in females. Okay, so mama dog, she's making eggs. Papa dog, he's making sperm. They're going to mate, and that enables reproduction. They produce the next generation. So 1 sperm, 1 egg. That's what I want to stress. 1 sperm, 1 egg. Now you have the zygote. You have the offspring. You cannot have 3 sperm fertilizing 1 egg. Doesn't work that way. 1 sperm, 1 egg. Sperm's got the little flagella to swim to the egg, gets to the egg, penetrates in, and then it blocks all the other sperm from getting in, and then you have your zygote or your offspring. Now in the case of those Rhodesian Ridgebacks, they had twins. Oh, check this out. Twins. Just like people have twins, just like lots of other things have twins. Okay? So mom produced 2 eggs. Dad produced 2 sperm. I mean, he actually produced billions and millions of sperm, but different sperm fertilized each egg. That's why these puppies, even though they're twins, are genetically unique. They're born at the same time, they were developing at the same time in mom, but they're genetically unique because they came from different eggs and different sperm. Now you can have genetically identical twins. That's 1 sperm, 1 egg, and then that starts developing into the zygote and it splits apart. But that's not usually the way twins work. Most of the time when we have twins, it's 2 eggs and 2 sperm. Genetic uniqueness to each individual. Now, yeah, there's a lot of similarities. They look very similar, maybe the same hair color, the same eye color. But that's simply because of the genetics carried in the eggs and the sperm. But there will be differences. So I want you to think about all the people out there that are twins that you know of. Friends, family members, etc. Sometimes twins are, well, you got a boy and a girl. Huge genetic difference there. Difference in the X chromosome. Twins, oh, you're tall, you're a little shorter, etc. Again, the key with meiosis is this genetic difference in every single egg and every single sperm produced. That's what gives the species diversity. That's what sets the foundation for evolution, is genetic diversity within a species. Why will some individuals survive while others do not survive? Okay, genetic diversity. All right, so now when we talk about people, and we look at humans, think about the human genome. What makes a human, or what makes up human genetics? Okay, humans have 23 pairs of chromosomes. I'm going to highlight and underline this. Okay, 23 pairs of chromosomes. That makes up our genetic code. And we're going to come back to this. All right, so everybody listening to the lecture, 23 pairs of chromosomes. Here's the example. So you got pair 1, pair 2, 3, 4, etc. The difference between men and women comes down to this 23rd pair, what we call the sex chromosomes. So that pair right there, colored in. Okay, if it's XY, that is a male. If it is XX, that will be the genetics for a female. So pair 1 for pretty much everybody on the planet is pretty identical. Same way with pair 2, pair 3, pair 4, etc. Our pairs are very, very similar. There are small, subtle, little differences between each individual. Within pair 1, there's going to be a couple little differences. Pair 2, there's a couple little differences, etc. Again, that's your genetic fingerprint. That's your genetic uniqueness. So when an egg and sperm is produced, it will contain, and this goes back to the process of meiosis, it's going to contain 23 chromatids. So you guys remember chromatids? Let me highlight and underline that one. Chromatids. A chromatid is half an X. There's a chromatid. You're going to have 23 of those sitting inside of your sperm cell. And over here, if this was the egg, you'll have 23 chromatids in the egg cell. Put those together, 23 chromatids, 23 chromatids. You got 46 chromatids. That's your genetic that's genetic information you need to produce a zygote. All right, so meiosis is all about how do we go from these 23 pairs of chromosomes down to 23 chromatids. How do we divide all that stuff up and mix it up and make it diverse and unique, and every sperm produced has just uniqueness to it. Well, what makes this diversity, what produces this uniqueness, is an event called crossing over. So I'm going to go bold and make this in red, and we're going to underline it. Okay, crossing over. This event occurs during prophase 1. So the way this works, in the female, all of her chromosomal pairs, you come back here, will buddy up. So pair 1, pair 2, pair 3, pair 4, etc. And randomly, these pairs, pair 1, this chromosome will overlap with the other one in pair 1. Come back to this. Okay, so if we have chromosome pair 1, there's your chromosome. Maybe the top part overlaps. So right up here, this is crossed over. Now on pair 2, maybe the overlap is on the bottom part. So crossing over occurs on pair 2 down here. Let's go pair 3. Maybe pair 3 got kind of crazy and crossed over top and bottom. So now you got 2 spots for crossing over. Right there and right there. Again, this is completely random. But think about how many different possible combinations you can create with crossing over. So hypothetical. Pair 1 is the only pair to cross over this month when mom's making an egg. Next month, pair 2 is the only one to cross over. The following month is pair 3, and then pair 4, and then pair 5. So right there, if you only cross over one spot on one chromosome, you have 23 different possible combinations. That's 2 years of reproduction for an average female, or 23 months of genetic variation. Now let's go if 2 chromosomes cross over. How many more months of genetic variation? What if 3 chromosomes cross over? What if 2 chromosomes cross over at 2 spots? This is why it is statistically impossible for anybody to produce 2 eggs that are identical or 2 sperm that are identical at any point of their reproductive lifespan. Because keep in mind, meiosis is a window of reproduction. It starts in puberty and it runs until we get through menopause. It doesn't go our entire lives. But the crossing over is what produces genetic uniqueness. Okay, so, get my spelling right. Alright, so crossing over is, there we go, is what produces genetic variation within every single sperm or egg produced. Okay, that's crossing over. Okay, incredibly important event. This is happening in your body as you're producing your eggs and your sperm. So then if that egg gets fertilized by a sperm, that's going to be genetically unique. And that's your genetic fingerprint. This is why DNA evidence is so important when we deal with crime scenes and things like that, because you have your own unique genetic code. And it's pretty hard to say it wasn't me, that's not my DNA at the crime scene, it's somebody else's DNA. Alright, so when we look at meiosis, meiosis has 2 parts to it. Meiosis 1 and meiosis 2. Okay, that's it. Meiosis 1, meiosis 2. Each meiosis goes through prophase, metaphase, anaphase, and telophase. So it's going to have a lot of similarity to the cell cycle and mitosis. The order of events is the same. There's a lot of overlap with what happens during these stages that will be the same. But the end result is what is most important here. At the end result of meiosis, when it's all said and done, you have 4 genetically unique cells. In the cell cycle, you had identical cells. 1 turned into 2, and they were identical. In meiosis, 1 turns into 4, and they are genetically unique. And then they're done. They either get reabsorbed back into the body, they get shed during menstruation. Those 4 cells are gone. So we all have a basket of cells that will go through the meiosis process, and when our basket is empty, we're done with meiosis. We're done with reproduction. For females, for human females, generally in your late 40s, early 50s is when you're going to be done with meiosis. And for males, usually later 50s into your 60s is when there's that decline of sperm production. Eventually, I said, we're all done with the process. And we're finished. But the cell cycle keeps going. Might keep going for another 20 or 30 years until the day you die. So the end result of meiosis, again, is 4 cells. Now, when we look at meiosis 1, the end result will be the production of 2 genetically different cells. Each cell will contains 23 chromosomes at the end of meiosis 1. Okay? So what we're going to do in the next lecture is look at what goes on during meiosis 1 and then meiosis 2 and problems that may arise.